Pharmapal.
Pim1 structure and innovative anti-cancer drug design. Welcome back to our channel. Today, we're diving into some exciting research on Pim1, a protein that could change how we treat prostate cancer. First things first, what exactly is Pim1? Pim1 is a serine/threonine kinase, which means it's a type of enzyme that helps regulate various cellular processes by adding phosphate groups to proteins. It is known to be constitutively active, meaning it works continuously without needing to be activated by other signals. Pim1 is often found in higher levels in various cancers, including prostate cancer and some blood cancers. Now, let's talk about the findings from the study. Monoclonal antibody treatment. Researchers discovered that using a Pim1-specific monoclonal antibody could suppress the growth of prostate cancer cells in both lab and animal models. This is groundbreaking because it suggests that targeting Pim1 could be a viable treatment strategy for prostate cancer. RNA interference. Another part of the study showed that when Pim1 was depleted using RNA interference, the proliferation of both mouse and human prostate cancer cells decreased significantly. Importantly, knocking down Pim1 did not affect the levels of its relatives, Pim2 and Pim3, indicating that targeting Pim1 could be specific and effective. Understanding the structure of Pim1 is crucial for drug development. Let's break it down. Pim1 has a two-lobe kinase fold. The N-terminal lobe is mainly made of beta strands. The C-terminal lobe is composed of alpha helices. The ATP binding pocket is located in the hinge region between these lobes, which is essential for its activity. Here's where it gets interesting. Over 400 small molecule inhibitors targeting Pim1 have been identified, with many showing promising results in inhibiting cancer cell growth. These inhibitors work by inducing G1/S cell cycle arrest and apoptosis, which is a programmed cell death that can help eliminate cancer cells. However, there are challenges ahead. Despite the promising data, no Pim1 targeting drugs have been successfully launched yet. One inhibitor, SGI-1776, showed potential but was withdrawn due to serious side effects like cardiac issues. The good news? New inhibitors like SGI-9481 are being developed with improved properties, aiming to reduce side effects while maintaining effectiveness. Next, we'll discuss the key figures and tables from a pivotal paper titled Insights from Pim1 Structure for Anti-Cancer Drug Design. So, grab a cup of coffee, sit back, and let's explore. Let's start with Figure 1, which showcases two-dimensional diagrams of the interactions between different inhibitors and the ATP binding site of Pim1. A BAM1, PDB code 1XWS, forms a hydrogen bond with Glu-121. BK00135, PDB code 2C3I, interacts with Lys67. C. PLXK013, PDB code 1YXX, establishes direct hydrogen bonds with Glu-121 and ASP186. D. The phenylpyrimidine derivative, PDB code 3DCV, forms two hydrogen bonds with Glu-121 and Lys67. These interactions are crucial because they help us understand how these compounds inhibit Pim1 activity. Next, we have Figure 2, which provides a structural overview of the Pim1-AMP-PNP peptide complex. The ATP binding pocket is highlighted, showing how the P-loop and hinge region contribute to binding. The hydrophobic areas are marked in yellow, indicating where the inhibitors can effectively interact with Pim1. This structural information is vital for designing more effective inhibitors. Now, let's transition to the tables presented in the paper. Table 1 summarizes the IC50 values of various Pim1 inhibitors. As you can see, SGI-1776 is a potent inhibitor for Pim1, with an IC50 value of just 7 nm. This table helps us compare the effectiveness of different compounds quickly. Table 2, Clinical Studies and Adverse Effects. Moving on to Table 2, which details the outcomes of clinical studies involving SGI-1776. The table notes that while SGI-1776 advanced to Phase 1 clinical studies, it was withdrawn due to QTc prolongation, a critical side effect. This highlights the importance of monitoring adverse effects in drug development. In conclusion, the research on Pim1 is paving the way for new cancer therapies. By focusing on its unique structure and function, scientists are hopeful that we can develop targeted treatments that could significantly improve outcomes for patients with prostate cancer and other malignancies. Thanks for watching. If you found this video helpful, don't forget to like, share, and subscribe for more updates on the latest cancer research and treatment innovations. See you next time.
Pharmapal.